Welcome to Ream Library. My name is Tom Laney. I direct the McFarland Center for Religion, Ethics, and Culture. Throughout the year, the McFarland Center sponsors and supports programming that explores meaning, morality, and mutual obligation. In addition to our ongoing lectures and fishbowl discussions, each year we pick a theme to explore through lectures, conferences, and other events. This year, the theme is the practice of science in a world of competing values. The goal of the series is to get students to think about ethical challenges of doing science today, managing expectations from government, from corporate funders, thinking about how scientific work is prioritized, thinking about research among animals, a variety of other issues, or involving animals, other issues. Scientific work provides at least as many opportunities as there are challenges, and tonight's speaker to open the series is helping us to think about both in a cutting-edge field, a field that he's helped invent. As many of you know, Holy Cross has a really remarkable tradition of accomplishments in science. The list of Holy Cross alumni with leading roles in the sciences is impressive. Dr. James Shannon of the class of 25, who was the first director of the National Institutes of Health. Dr. Anthony Fauci, the class of 62, known for his leadership of the National Institute of Allergy and Infectious Diseases. And of course, Dr. Joseph Murray of the class of 1940, a Nobel Prize winner for his pioneering work with human organ transplants. Tonight's speaker, Jim Collins, is doing work that puts him in a class with these scientists. And perhaps on the track, we hope to maybe a second Nobel for Holy Cross. We'll see. Maybe the third Nobel is right here in our audience. I have to hope for that. So, Jim Collins is a member of the class of 1987, a pioneer in the emerging field of synthetic biology. He's the William F. Warren Distinguished Professor, the University Professor, and Professor of Biomedical Engineering at Boston University, where he directs the Center for Synthetic Biology. He's also an investigator at the Howard Hughes Medical Institute and a core founding faculty member at the Wyss Institute for Biologically Inspired Engineering at Harvard University. At Holy Cross, Jim was a physics major. I told Janine, see, this means that you can do something with your life if you're a physics major, right, Matt? He was a runner, and he was class valedictorian. He met his wife here, the likewise accomplished Dr. Mary McNaughton Collins. He went on to a Rhodes Scholarship and earned his doctorate in medical engineering from the University of Oxford. Jim has been awarded a MacArthur Genius Award, a National Institute of Health Directors Pioneer Award, as well as the Metcalf Cup and Prize, BU's highest teaching honor. He's been named to the Technology Review list of 100 top young innovators and the Scientific American list of 50 outstanding leaders in science and technology. He's an elected member of the National Academy of Engineering, the American Academy of Arts and Sciences, the Institute of Medicine of the National Academies, and the National Academy of Inventors. In addition, Jim's been involved in a number of startup companies, and his inventions and technologies have been licensed in several biotech and medical device companies. He's also been generous to the college as a trustee, but recently finished his term and passed that along to some other people. Please join me in welcoming Jim for his talk, Synthetic Biology and Redesigning Life, Hopes and Challenges. Thanks, Jim.
I want to thank Tom for the really kind introduction, but even more so for the opportunity to start this marvelous lecture series. And what I want to do over the next 45 minutes or so is really just have some fun and give you a sense of what is happening in this new field called synthetic biology. And in the context of it, also provide for the students, it's so nice to see so many students out here on a weeknight, what it's like to be a scientist, a bioengineer, and how your career really responds to unexpected and random events. So this field that I'm talking about is really only about a dozen years old. And it's this relatively new group of scientists that are bringing together engineers and molecular biologists to design synthetic gene networks and to use these to reprogram organisms. So I want to give you some background on myself of how did I get into this and how did I help launch it. And I, as Tom said, studied physics here. I actually was brought up by engineers. And I intended to study engineering in undergraduate. And in the midst of visiting college, fell in love with Holy Cross and chose in the end between MIT and Holy Cross. And in fact, it was one of the best decisions I ever made in my life to choose Holy Cross. Sorry, thank you. That was unmuted. And it exposed me to so many different sciences. And when I was here as a student, I had the opportunity as a Fenwick Scholar to do a year-long project. And I did a project with Ken Prestwich in biology, wherein we focused on functional electrical stimulation. This was a relatively new technology back then that involved the application of electrical currents to the muscles of paralyzed individuals with the hope of restoring functional movement, say, for example, walking. I carried on related work at Oxford in biomechanics, and when I joined Boston University in 1990, we shifted toward applying principles from physics and engineering to whole body dynamics around balance. So we studied how people maintain balance, but also studied how could we improve balance, and came up with some odd notions, including the idea that you could have vibrating insoles. So we showed you could deliver low amounts of vibration in the soles of subjects' feet at levels they couldn't feel and improve their balance. Most stunningly, we could take a 75-year-old and have them balance like a 25-year-old. And in fact, I gave a lecture in this hall probably now 8 years ago that I think was entitled Building a Bionic Jesuit. And so we have not yet done so. But the question is, how do you go from building a bionic Jesuit to an engineer playing around in molecular biology? And so the story goes to about the mid-90s. When I joined BU in 1990, I actually joined on the research faculty. This had the downside that I didn't teach, and I had continue to have tremendous respect for the teachers I had here, the huge impact. But it had the upside that I never had to go to a faculty meeting. Well, at one of these faculty meetings, Charles Cantor, who was then our chair, had hatched a scheme where they were going to propose to the Whitaker Foundation, which was a new foundation then giving out millions of dollars to launch bioengineering programs. And back then, molecular biology had no position, no place in a bioengineering department. And Charles and the other faculty decided they were going to basically prepare a new training program that would take engineering students from genes and proteins to cells, to tissues, to whole organs, to whole bodies. Now, this is the standard de rigueur around the country in 2013, but in 1995, nothing was happening. The department wrote up their proposal, sent it in, they made it to the final round, and they were going to be site visited. And the Whitaker Foundation liked to see early efforts on what had been pitched, including, say, bridging these gaps. So Charles Cantor called me into his office on a Monday, and he says, Jim, we're going to be a little embarrassed. We have nobody bridging these gaps. Could you take your physics principles, the engineering principles you've been applying to the body, and apply it to a problem we have down at the molecular level? I said, well, I know nothing about molecular biology. He says, that's okay. I said, okay, if you take that, let's see what we can do. So he says, I've got a system for you. We've engineered a cell-suicide system that's designed to gobble up stuff in the environment and then kill itself. Except it's killing itself before it gobbles up all that it should be gobbling up. We want you to make it tighter. Could you model this in control? So maybe. He says, okay. He goes to his whiteboard. He writes down these equations. I write it down. He says, see what you can do, Jim. As I'm leaving his office, he says, so Jim, you have 4 days to come up with something. The site visit's Friday. So we went rushing back to my lab. No molecular biologists in sight. And for the math-oriented here, there's a few, there were no interesting dynamics to those equations, and they had little, if anything, to do with what Charles wanted us to model. We didn't know enough to modify it to come up with something. So I went to Charles on Thursday night. I said, Charles, we're going to have to punt. I tried, he says, no. He says, I want you to go out there tomorrow and give them your vision for what an engineer can do in molecular biology. I said, Charles, I don't have a vision. You gave me this assignment. He says, no, go out there, we're going to have. So they wheeled me out the next day. I was a junior faculty member, gave my vision, and we did not get that grant. Millions of dollars lost, and it's actually a famous site visit that's known around the bioengineering community, and I played a very big role. Well, when we now sat back and had more than 96 hours to think about molecular biology, my students and I got very excited as we saw the possibilities. Now, this is 1996, so it's around the time that I think some of you maybe just were born. And the #1 science project in the States was the Human Genome Project. So this was a massive, multi-billion-dollar program to sequence multiple organisms, including humans. Very few engineers were involved in that program. And Charles and my dean, Charles Lissy, who were involved in this, said, Jim, go talk to the leaders. So I talked to Eric Lander and Lee Hood, and he says, Jim, we need your engineers. We have these enormous parts lists coming out now where we need to figure out how those parts operate in what we'll call circuits or networks or pathways. And it's those operations that underlie interesting biology and disease. And I said, okay, great. What do you have for data? Well, it turned out they have these things called gene arrays, which some of you may have worked on or studied, that were just on the market then that allow you to assess how genes are being turned on and off inside a cell. Turned out they're very expensive back then, still are. There were only 7 publicly available data sets. So the idea that you could reverse engineer or infer the structure of a network with that bit of data to give you perspective would be as if you're going to solve a 3,000 by 3,000 Sudoku puzzle with only 7 numbers filled in. Right? Impossible. You'll get all the numbers you want, but you won't come up with what the creator had. So Tim Gardner and myself ran as fast as we could away from that problem. And to give a perspective for the young people, particularly for the parents in the audience, as you know, there are 2 kinds of kids who become engineers. You have the kids that take stuff apart, right? They take apart your lounge chair, they take apart your radio. Rarely do they put it back together. But in this age of self-esteem being preserved, you never scold them, you applaud their curiosity as you try to put back, right? Those are the kids who would have tried to reverse engineer the natural cell. The more common kind of kid that becomes an engineer are the tinkerers. So the tinkerers are the kids that put stuff together, right? So in my day, it was an erector set. It were Tinker Toys. It was the old Legos. Nowadays, Legos is the other kind of set scheme. And so Tim Gardner, my student, star student, who was an engineer at the time, he and I sat back and says, why can't we do this in molecular biology? So we took inspiration from electrical engineering. We could come up with circuits. You could do modeling. And then just as an engineer would go find parts, transistor, resistor, capacitor, we would go find molecular parts, genes, bits of DNA, turn those genes on, bits of DNA to shut off those genes, put them into plasmids, using schemes I'll talk about in a second, get in a cell and see the functions. Well, we sat back and explored over 9 months or so what you could do in that space and considered a number of different circuit models motivated by electrical engineering and zoomed in on what's shown here, which is a genetic toggle switch. This was motivated by toggle switches or RS latches or flip-flops that are simple circuits in electrical engineering. What are those? Those are circuits that can be flipped between a 0 and 1 or an on or off state by transient pulse. It's a simple memory element that actually forms the memory for the devices that you all carry around nowadays. So your iPhones, your whatever the other brand, Samsung, you have hundreds of thousands of flip-flops in those devices. We sat back and said, how would you do this in molecular biology? And we came up with the following scheme, and I'm just going to go through it at a high level. But you basically have 2 different genes that you set up so they always want to be on. These are called using constitutive promoters. So you'll have your strip of DNA where these guys each want to be on, but you design it where they're trying to shut each other off. So a gene, when expressed, will produce a protein. We design the circuit in the abstract so that protein from gene A would bind to the on switch. So not right, but the protein from this gene would bind to the on switch for that gene, shutting it off. And the protein produced by this gene would bind to the on switch for the other gene, shutting it off. Okay? Each wants to be on. Each is trying to shut the other off. Right? So it's, again, for the parents, if you have 2 kids in the back, each want to talk. Right? That's an unstable situation. You shut up. No, you shut up. I want to talk. I want to talk. Right? You can settle it with an inducer, you're the parent, where you can set up so it's in one of 2 stable states. Either gene A is on and gene B is off, or gene B is on and gene A is off. Okay? And you can now flip it between those states by delivering a chemical or environmental stimulus to now shut off the currently active gene. Very nice in the abstract, and we got very excited. Well, I'll just show you one set of equations that I'll walk you through. Tim was a mechanical engineering student, and we started where it was easy to do. We didn't have any money to do this, which was modeling. So using differential equation type modeling of the sort I learned with Professor Schertzer in my math methods in physics, still one of the best courses I ever had that has strongly impacted the teaching I do at BU. You can take 2 differential equations, just briefly, where this is a rate equation. So you're studying how something's changing with time. This is the concentration of protein from the first gene. This is the concentration of protein from the second gene. You've got different terms that you can then say, okay, what happens in the steady state when they're no longer changing in time? From the equation, that means you're going to set these equation of change time to 0. So if you do that, you can now study these that call nucleins, where in the protein space of the 2 proteins, you can see how their dynamics in the static state are changing, such that you can have one where they're both on, this is unstable, or you've got it state 2, gene B is on, gene A is off, or state A, state 1, gene A is on, gene B is off. We can do many, many more slides of these than we did to basically convince ourselves this could work. Now, this is around 1999. Tim and I had no molecular biology background, and we went and talked to about 10 molecular biologists. And this is 1999. We had transparency, so printouts of these. And the conversation with almost everyone went as follows. We'd share the results and says, what do you think? Oh, very interesting, guys. Do you think it can work? No. And we pressed them. 2 to 1. They said no. Why don't you think it can work? And they said, well, we think you're going to get the biostability, you really have to have tight off, and it's very hard to get tight off in a cell. 1, 2 is you're going to have to make these very big cells, don't like having a lot of stuff in. They're just going to spit it out. They went on, and invariably, my molecular biology colleagues would kind of pat me on the head as we left and say, you know, biology is complicated. You should stick to engineering. Well, we didn't get too discouraged. We went back to Charles Candor. Says, what do you think? He said, I got two answers for you guys. One is, crazy enough idea, I think it'll work. And two, you two are so naive of how hard it's going to be, I bet you two can get it to work. And with that ringing endorsement, he opened up his lab to us, which I think he has since regretted. We've metastasized to about 20 benches. And what we did was try to build circuits in E. coli. So this is the kind of things you hear about scares on Brussels sprouts or hamburgers. In labs like mine, we use the neutered kind. They're safe. You don't want to eat your experiment, but you're fine, provided you wear gloves. And what we did was find parts out, different genes, different motors. And what you have in molecular biology are enzymes that can serve as chemical scissors. So Professor Mary Lee Ledbetter here, I took a genetic engineering course with Professor Ledbetter in 1985, where she taught us about recombinant DNA. And oddly, 15 years later, I used what I learned to help make these, where we can cut these together into plasmids and get them to function. And just briefly, this thing in cells worked beautifully. You can start it in the off state, flip it on, it goes to on. You can keep it in on, flip it from on to off. So in flipping on, in this case, we're using a chemical, a sugar to do it. Flipping it off, we're giving heat shock. And we've developed both chemical switches like this, as well as chemical temperature switches, all of which are stable. Flip it on, this thing can stay on, keep going. Well, it turns out this has an incredibly sharp switching threshold, which you might need for applications I'll bring up in a second. Whereas you're sweeping a scheme, this is in a true off, and then over a narrow range of whatever chemical you'd have it for flip or sense, it actually flips from off to on. Well, it turned out that this thing had an incredibly long switching time. It took about 6 hours to flip to the on state, and there are many molecular reasons for that. It was on a high copy plasmid for the life scientists in the audience. But it had a faster, but still nonetheless slow, 25-minute to flip it from on to off. And there, with heat destabilization, you could have the protein that was keeping the guy off. It destabilized with heat and falls off. So what did we do? Well, in this case, to really help this field get launched, we basically had engineered cells with a new circuit they had never seen before, where we could endow them with memory, making now the cells to become programmable. So this was an ad you can't see so well, but it has fluorescing cells that Accenture used to launch when they were spinning out from Arthur Andersen, one of the best business spin-outs in American history. So shortly after they spun out, Arthur Andersen imploded from the Enron crisis, which had some contribution from the Holy Cross grads, and Accenture continues to go along quite well. Well, they had these fluorescing cells, then popped up. Bacteria tested as digital circuits. Now it gets interesting. And in fact, it was based on our work. So you had this idea now that you could program bacteria with memory and other elements. And back in 2000, or again, I have the students here, so you all were maybe just starting elementary school, one of the big excitements around the country was actually biocomputing. The idea that you could program cells to behave as a computer. And in fact, when I first presented this, it was at a DARPA workshop on biocomputing. As I went through the slides, the audience was like, yay, the BU guys have done, woohoo, we have a genetic toggle biocomputing. And I said, okay, wait a second, everybody. I said, this thing takes 6 hours to flip. All the air went out of the room. And why? Well, the machines you carry around, those flip-flops take tiny, tiny fractions of seconds to flip. So no time sooner you're going to have a computer with E. coli inside to do different things, including surfing the web, right? Imagine you're going to surf the web with your bacterial computer. Well, 4 days later, when weather.com finally comes, they say, oh, I didn't realize it was supposed to be 83 degrees at Worcester and 110 degrees in the REM library on Wednesday night. Okay. Well, when you think about synthetic biology, this is the wrong way to think about biocomputing. The better way to think about it is an area that's incredibly rich in the field, and that is the idea of programmable cells. Can you use this engineering approach to the interface of molecular biology to create circuits that you can now insert into cells to couple with their already beautifully evolved input pathways or beautifully evolved output pathways to do cool stuff? And this is work that we got very excited about and continue to do work on. And the thing we got most excited to start in this work with Hideki Kobayashi and Mads Kern in my lab was to create whole cell biosensors. So could you program cells to sense stuff in the environment? Could it be a DNA damaging agent, which is what I'm showing here? Could you program cells to sense pathogens? Is there an anthrax? Could you program cells to sense heavy metals? Is there lead coming in on an import from China? Well, we showed you could do this. I won't go into details, but we could take our switch, couple it to natural sensing pathways inside bugs, and have it now beautifully sense DNA damage. One thing you should appreciate is, from a sensor standpoint, you always want to have low false positive rates. What does that mean? You don't want it going off unless you sense something. So you have anthrax outside Hogan, you only want that going off if you have anthrax, right? And the way you can relate it is we all have sensors in our dorms, in our offices. You have smoke alarms. And you only want that smoke alarm to go off if there's a fire of this smoke. This had beautifully stable off states. 48 hours, 96 hours, these things wouldn't flip. So you can imagine having them outside Hogan or inside somebody's body. But they would flip, in this case, this is now 9 years ago, with a higher sensitivity than what was then the best available DNA damage sensor. Well, a colleague of mine, Ron Weiss at MIT, took these switches and coupled them to signaling pathways in bacteria. Bacteria can actually talk to each other via quorum sensing signals. He was able to engineer intercell signaling systems to have bacteria, both as senders and receivers, couple it so that they could talk to each other and actually form bullseyes around the source of whatever you want to detect. And he's beautifully shown this. And you can think of then many interesting applications, including, for example, using bugs, microbes, to identify mines in an abandoned minefield. Related to that, Ron and I have a multi-million-dollar grant from the Office of Naval Research to do just that, coupling these guys to mini robots and using it to sense explosives in the marine environment. So the idea went far away from getting anything deployed in a marine environment. But the notion would be you could have these to go and sweep a harbor before you actually had deployment or a cruiser go in. Well, this is in the McFarland Center that focused in part on ethics. So you can imagine when you start thinking about molecular biology coupled with engineering, for those who haven't neared heat exhaustion at this stage in my lecture, can begin to think about all different crazy scenarios. And I can share with you that this field is rife with interesting ethical discussions. And much of it leads to controversy. They're worried that the engineers are mucking around in molecular biology labs, and that we don't know what we're doing. And so they're worried that somebody in my lab is going to create a Franken microbe that's going to escape and, God forbid, eat Fenway Park. Well, as if the hype couldn't get any bigger, and to give you a sense of the level of hype, there are many, many more magazine articles and newspaper articles about synthetic biology than there are original research reports. So for the academics in the audience, you can appreciate that the scales are just imbalanced. And it captures people's imaginations, and it captures their concern. Well, as if the hype meter couldn't go any higher, Craig Venter, who was the commercial guy behind the sequencing of the genome, entered the field in May 2010 and reported, this isn't Craig, this is a magazine cover about Craig's work, that he had created a synthetic cell. And I'll explain what he did, and I'll show how the media presented. What he had done was he had synthesized the genome of a natural bacterium, mycobacterium, sequenced it, he then synthesized it and transplanted it into a related bug and got that bug to take up that genome, replicate, and behave as if it was like its cousin that got the transplant. Beautiful work, but the media picked up as if the first artificial organism has been created. Man has created life. And of note for Craig, President Obama and the Pope both commented on the same day on his work. Peer review I've never had, and I hope to never have. Well, you can imagine that this created a lot of concern and a lot of discussion, and I'll come back to what President Obama did in a few slides. But we engaged in the discussion, and I'm happy to, in the question period, to open up interesting related questions. But we also took it on as engineers and said, okay, if there's concern about this, that something escapes from our lab and is dangerous, could you hit it head on? Could you engineer safeguards into these bugs? So could you engineer, for example, circuits that would program cells to count so that if they're out in the environment or they escape or they're put in your body, that after some period of time, they're programmed to commit cellular harikari? Well, what do? We created cells that could count. We created circuits shown up top that I just walked through. They're basically daisy chain circuits where you have a switch that's in an off state. It has to see a pulse, maybe a pulse of sunlight or a cell cycle protein. It flips on, primes then the next guy that had been off to respond to the next pulse, et cetera, et cetera. So it's as if you're going to run down a hallway, opening doors as you run down the hallway, and you need to open all those to get to the end. Well, we showed you could do this to program cells to count to 2, to 3, to 4, et cetera. Tim Liu, a student of mine, picked up on Ari Friedland's work that was prior to that and actually created what are called recombinase switches. So what he could do is now program very similar daisy chain, but you can express enzymes that will take a chunk of DNA, take it out, flip it, and reinsert it. And he set it up so that when it's in its original unflipped state, the gene downstream doesn't flip on. When it gets flipped, the gene downstream gets flipped on, primes next, et cetera. This thing worked beautifully. Shown up on the corner here is this guy could beautifully count to 3 and higher. Interestingly, the first guy operated on 20 to 40 minutes. You'd couple it to cell cycles. And this guy operated 10 to 20 hours. So you could envision having this coupled to light-dark cycles. Could you program these guys to die? Well, another student of mine, Jared Kluwer, did just that. He created a circuit that has a logic gate, so-called AND logic gate, where it has to see two signals. If it does, it will flip on a gene, a pair of genes in this case. They will produce proteins that will rapidly kill off the bug. Two interesting anecdotes to this. One is that President Obama, on hearing Craig Venture's announcement, charged his bioethics commission with their first task, which was go check out those synthetic biologists to see do they know what they're doing and do we need more regulations that go beyond what we learned in Professor Ledbetter's class that came out of the Asilomar conference in the 70s. Well, 2 points. That one is they concluded, no, we don't need additional regulation just yet, but we'll keep an eye on this field in case they get a little crazy. But two, they actually highlighted this as a much-needed safeguard. And if you're going to be doing these things, putting them in people's bodies, putting them in the environment, even playing with them in your lab, you better have safeguards should you have an escape or need to get rid of them. And there's now multi-million-dollar programs from DARPA. Two, and more interesting, when we published this work, I got called by a number of biotech companies. Says, Jim, we want to work with you. We want to use your switch to protect against corporate espionage. I said, oh, do tell. This sounds intriguing. He says, what's up? And he says, well, here's what we do. We engineer bugs to produce interesting things. So, for example, one company called they're engineering bugs to take sunlight and make fuel. And he says, we're going to probably put in about $500 million into the development of this highly optimized bug. We're worried that the summer intern we bring in is not really a summer intern, but is a spy from a corporate adversary or an enemy of the state. And we're worried that at night the corporate intern will take a jar, go into the big reactor that has these bugs going up to a certain density, scoop out the secret sauce and run out the door. And so they've challenged us, and this was from multiple companies, to program these bugs so that if they're taken out of the reactor, they explode. I said, okay, and we actually know how to do that very well. They said, but we want you to go even further. We want you to have them shred their DNA when they explode. I said, okay, I said, I don't know how to do why. And they said, well, we're worried the bad guys might have Craig Venture tied up in the back. So remember now, Craig was the guy who could synthesize a whole genome. And they said, figuratively, that you've got somebody trained by Craig so that if your cell explodes, you can still fish out your DNA. Some of you have probably done these experiments. Right? So now if you fish it out, the secret sauce is not in the bug. For the most part, there may be interesting ribosomal changes, but for the most part, it's encoded in his DNA. So the thought is, you better get rid of that so they can't put it back together because they could put together the bug. Well, we're not working on that, but we are working on these exploding bugs. What I want to shift now is for the next few examples is away from biotech and toward medicine. And interestingly, about 2 years after Tim and I came up with the switches, we turned back to the original problem that got us into this, which was this idea, could you reverse engineer natural circuits? So this was 6 to 7 years later. By now, there were hundreds of data sets. And Tim and I realized that we could take our switches and use them as probes into natural cells. And again, interestingly, inspiration came from electrical engineering. So in electrical engineering, you'll have techniques where you can actually infer the structure of a circuit. So for example, if you're an engineering undergraduate, electrical engineering undergraduate, invariably you'll take a circuit-based course where the professor will hand you a circuit board. So these are the green boards that I showed you inside the bug on the first slide, but you'd find it if you break open your machines. And they'll say, okay, hotshot, how is that wired up? And they don't give you the wiring diagram. What do? You run your electronics lab, and you turn as many of the components as you can up or down or on or off. You measure the response of those, and then what you use are basic computer algorithms called machine learning algorithms to infer the underlying structure. You then run back to the professor, show them what you have. If you're a student at Harvard, you get an A no matter what you actually show the professor. If you're a student at Holy Cross, we use the full spectrum of letters, as we also do at Boston University. We realized you could do that inside cells. Now to give, again, there may be a few with the 3-2 program. I assume we do still have 3-2 program, Frank? Columbia or WPI? Oh, one of them. So most of you are not engineers, but many of you own a home or lived in a home. If you buy an old home, you never get the wiring diagram for your house. But invariably, you'll have a circuit breaker box in the basement. Invariably, it's not labeled. So what do you do to label your circuit breaker box? You reverse engineer, just as we did. You go upstairs into your kitchen, you take your boom box, put the volume on high, you plug it in, you run downstairs, you flip the switches till you hear the boom box go off, and now you label that kitchen. You run upstairs, you plug it in the dining room, you run downstairs, etc. You get a first-order model of your house. That's exactly what we did here, where we worked out means where now genes or proteins, we can tell you which gene is talking to which gene. We had dinner, somebody's working on protein-protein interaction, so we can tell you protein to protein, gene to gene, metabolite to gene, etc. We can tell you the direction of the interaction, the nature, is it active inhibitor, and the strength of the interaction. And that latter is critical for another application we came up with, which is we recognized that you could use this to go after the targets of drugs. So what happens in this target-rich age is that somebody will identify an interesting protein or gene that needs to be inhibited. Biotech and pharma companies are incredibly good at setting up a chemical assay to see, out of all these 100,000 possible compounds, which ones hit that target, and thus you then develop that guy. Really good at doing that, and some of you may have worked for companies that do that. What they're really bad at is telling you what else do those compounds hit. And you have thousands and thousands and thousands of possibilities in any given cell they're targeting, be it bacteria, fungal, or mammalian. I maintain, and it's not particularly original, that most drugs fail because they hit a lot of stuff. I maintain, which is more original, most drugs are effective because they hit a lot of stuff. Well, we realized you could use this to go after it, and now the inspiration came from control engineering. I teach control engineering to our biomedical engineering undergrads, so it's a core course. I get the whole crop with them. From lecture 1 to lecture 27, I emphasize two of them, that there are basically 3 parts to a control system: the input, the output, and the system itself. If you know 2 of the 3, you can work out the 3rd. Very simple notion. But now imagine you've got a drug where you don't know what it's hitting. You have your network model of whatever organism you want it to hit. It could be a human cell. It could be a bacteria. It could be a fungi. If you can measure the output, you can filter that through that network and identify what likely was the input that caused that response. Very simple notion. And we got excited, showed you could do this in silico. We've done this for mammalian systems. We've done this for fungal systems. But what my lab is primarily focused on is bacterial systems and trying to develop better antibiotics. And the reason we got excited about 7 years ago about this problem is scarily captured in this plot. The left plot is indicating something we're all generally familiar with, which is the number of resistant strains is dramatically growing. This was a problem in the past limited to hospitals. I tell my students, worst place to be when you're sick is a hospital. Get out as fast as you can because these bugs are going to get you. Now the problem is these bugs are not limited to hospitals. They're on our playing fields. They're in our gyms. They're at hotels. They're in child care centers. And in fact, the problem is bigger outside hospitals now than inside hospitals. This nasty problem is unfortunately coupled with the problem on the right. And that is the number of new antibiotics being developed and approved has been dropping dramatically over the last 15 to 20 years, particularly the last 10, 5 years. And the reason being is that biotech and pharma are getting out of the business. I'm a fan of biotech and pharma. I don't slam these guys, but I will in a minute. And for the most reason, it's basically economic. Antibiotics you take typically for a 2-week course, relatively cheap. There's a lot of competition. It's going to still cost you $500 million to develop your antibiotic. It's going to cost you $500 million to develop a diabetes drug. So the drug company is saying, okay, let's go after diabetes, let's go after cancer, let's go to metabolic disorders, chronic conditions where we sell pills. We're going to sell them a lot of pills, maybe for the rest of their life. So they're getting out of business. What I do fault them on, I don't fault them for that focus, is they're also focusing on lifestyle drugs. Chronic condition. Most stunning fact that I know, there are more Viagra-like drugs in development than there are antibiotics in development, which is, I think, a stunning comment about our societal priorities. I'm a positive guy. I went to Holy Cross. I motivate my group by saying, okay, while some of us may die of a bacterial infection, at least we'll have a smile on our face. So with that as our daily motivation, we went after it, and basically what we could do briefly is collect hundreds of these expression profiles I alluded to with gene arrays, where we could study how genes in, say, E. coli were being turned on and off under different conditions. Literally over 1,000 of these, and we could then reconstruct the entire network inside the bug. Briefly, we then looked at different antibiotics. You have 3 general major classes of antibiotics. You have what are known as quinolones. This includes things like Cipro. So they are thought to kill by inhibiting gyrase that blocks replication, the cells die. You have aminoglycosides, like tobramycin or gentamicin, that are thought to kill by blocking protein synthesis, boom, the cells die. You then have beta-lactams like penicillin. We've all heard, I have a penicillin allergy, supposedly. Ampicillin. They block cell wall synthesis, and the thought is that's how they kill, and the cells die. What we discovered is that in addition to all of those, they are also triggering oxidative stress inside these bugs, and that's contributing a major role to the killing. So oxidative stress will damage a lot of stuff inside the body, inside a cell or inside the body, DNA, lipids, and proteins. And again, we're bioengineers, so we're going to exploit that. And what we hypothesized was that you could go in now, if every drug, antibiotic designed to kill a bug, damages DNA, could you make the antibiotic stronger by blocking the defense response of the antibiotic? So in this case, could you block its DNA damage repair? And what we show here is that's exactly what you could do. We knock out the start switch for the DNA repair response, and we could make these antibiotics 10 to 1,000 times stronger. Well, you're not going to knock it out genetically on a patient. That's crazy. But you could do it chemically. So we set up a screening assay of the type I shared with a group at BU that actually is run by John Porco, who's a chemistry grad of Holy Cross, and it's a natural products synthesis screening facility, as well as with the biodefense facility at Harvard. We looked at over 50,000 compounds and just briefly identified many. I'll focus on CB301. By itself, CB301 does nothing to the cells. They're happy. Nobody dies. Everybody's good. When you combine it with an antibiotic at a low amount, you can boost the killing efficacy of that antibiotic up to 1,000-fold. So our hope here is that these small molecules can be given along with the antibiotic to make supercipro, or superpenicillin, or supertobramycin. Well, we also went after a synthetic biology notion, wherein we looked at bacteriophage. I'll explain in more detail in my next slide what those are and how they act. But these are basically viruses that go after bacteria. They don't go after mammalian cells. And so with Tim Liu, who was a great student of mine, he came in and says, could we engineer these guys to infect the bacteria so they would express a protein to shut off the DNA repair, the defense system of the bug, and thereby make the antibiotic better? And briefly, 2 key points on this is now not only 10 to 1,000, these bacteriophage could make antibiotics 100 to 10,000 times more effective. Second, it could resensitize a resistant strain to the antibiotic to which it had grown resistant. We actually did animal studies to show that we could rescue mice that otherwise would die. And the interesting backstory of this, when we published this work, we got immediately contacted by the U.S. Army and Walter Reed Medical Institute. And they shared with us that U.S. soldiers coming back from Iraq and Afghanistan are really troubled by nasty skin wounds that have resistant bacterial infections. And the wounds, as a result, aren't healing. And it's basically being caused by acinetobacteria, which are endemic to the region of Iraq and Afghanistan, for which our antibiotics don't work. And so we're now, as part of a collaboration with Tim Liu, my student, engineering bacteriophage to try to boost the effectiveness of the stuff we have. Again, much of the theme here is, hey, we've got pretty effective antibiotics. They're getting less and less effective, but can we use engineering skills to make them better? Well, coming back to the phage, what we also got excited about was, could we use bacteriophage to go after bacterial biofilms? Anybody know what a biofilm is? What's a biofilm? So you know it. Now I put you on the spot. What's a biofilm? Yeah. So it's basically a community of bacteria. They actually surround themselves in an extracellular polysaccharide matrix. So this is a complicated matrix. And they attach themselves to a surface. So the plaque on your teeth is a biofilm. The gunk in your sink is a biofilm. The stuff on the side of a ship is a biofilm. Why a bioengineer is interested in biofilm is bugs and biofilms. 2 reasons. Bugs and biofilms are 1,000 times more resistant to antibiotics than bugs that are free swimming. And 2, biofilms that are from bugs love to grow on anything you put in somebody's body. So if it's an artificial knee, an artificial hip, a pacemaker for your heart, a pacemaker for your brain, which you may need at this point in my talk, the number one risk now is not the surgery and the general anesthesia. It's you're going to get a biofilm infection. Say, for example, in the hip, it's a big problem. I think 2 or 3% now failure rate in this country from biofilm infections. You get a biofilm infection on the artificial hip. What do? They open you up. They remove your hip. They clear your infection, which means you're going to sit in a hospital bed for 1 to 2 weeks with no hip. They put in a new hip. They close you back up. No, thank you. I'd rather go to a faculty meeting myself than subject myself to that. Well, so with Tim Liu, we came and took on this challenge bioengineering. We said, okay, what could we do? Could we re-engineer the phage to go after this? So now we've got our bacteriophage, and let me just walk you through quickly bacteriophage biology for those who haven't seen it. You basically have that little lunar module. It's going to dock on a bacterial cell, and it's going to blow its DNA into that bacterial cell. Now the bacteriophage can follow one of 2 paths. There's one called lysogeny, where it basically tucks itself in the DNA and it's quiet. Maybe it expresses some protein of the type we did in that first example. You really don't know it's there. It's eating a little bit of your stuff, but just hanging out. The example would be as if you have a nice house guest, right? Nice, quiet house guest. You know they're in your house, share a meal with them, but they're there, you kind of carry on. The other path is the lysis mode, lytic mode. In lytic mode, it's as if you invited college students to your house. What happens? They eat all your food, they reproduce, they explode your house. Okay, not college students not Holy Cross students at off-campus parties. But, yes, I've gotten many of those emails. I hope you're all going to behave this weekend. What we did was harness the lytic mode. What happens in lytic mode? The phage will infect the bug, make multiple copies of itself, and then it explodes the cell. so that these guys will now have the possibility to infect a boatload of other cells. The challenge in biofilm, this is done before our efforts, is they can't penetrate all but the top layer because it's a gunk that they can't get below. So we're bioengineers. We engineer these guys to express on their surface enzymes to break up that biofilm. And just briefly, it worked beautifully. Here's a gunked up film on the left, engineered bacteriophage treatment on the right. Looks like an infomercial, but it really was beautiful. Here's quantitative data. This is the untreated scheme. There's a lot of strong antibiotic ampicillin. You get about a 20% drop. Strong amount of active enzyme, 50% drop. Unmodified phage, pretty good, 70%. But we get 99.99% clearance of the biofilm with the engineered phage. Tim Liu came forward, and he also did a Trojan horse version where, in addition to expressing an enzyme on their surface, he also engineered them so they'd express enzyme to break up the biofilm inside, as shown here. And he got beautiful results, again, on the greater than 99.99% expression. Well, for those who are entrepreneurial in the room, Tim Liu and Mike Kerr, a student of mine, launched a company around this technology. And there are interesting ethical and societal implications in what happened. They initially went after every university business plan competition that they could. So typically research universities, business schools, they'll have these competitions where you can win some money if you have a good plan. They won boatloads, raised probably 170, 190 million for their company. Pretty good, but that's not enough to really launch a biotech company. Their business model, was to use engineered phage to go after problems in infectious disease. They talked to the venture community, the investment community, and the venture community says, we don't want to go there. Yes, it's a big societal problem, but it's going to take us too long to get there. The regulatory hurdles to the FDA are too high. They said instead, go after industrial biofilms. So basically any system, any industry that involves bugs, living organisms, has a biofilm problem. And others, HVAC systems. So you have HVAC systems, air conditioning systems, biofilm fouling. The Legionnaire's disease outbreak in the mid-70s in Philadelphia was the biofilm fouled air conditioning. Oil gas pipelines, paper mills, et cetera. These guys went out and raised multi-million dollars to go after industrial biofilms. They were very excited. They built a team of 20. And they went to the EPA. They had gone to the EPA on the basis the EPA says, okay, no problem, guys. 2 to 3 months, we'll give you approval. We're going to give you blanket approval for your cocktail of phage needed. Biofilms in the real world, not in my lab, but on the top of a building or in a bioreactor, have many, many different bacteria in them. So you're going to have a cocktail of phage. Well, that was really bad advice they got from whoever they talked to the EPA, because when they went back with now real samples, the EPA said, oh, no. You're the crazy engineers. You're engineering these schemes. No. You're going the 18, 24-month route, and every time you change one of those guys, we want to see you again. And so the company sat back and says, no, okay, we can't get there. It's too long. And they sat back and did an amazing pivot. They've changed their name from NovaPhage to Sample 6, where now they're using engineered phage to go after and detect pathogens in the food industry and in hospitals. So things like E. coli, listeria, salmonella, big problems. And what happens at food packaging plants, production plants, shipping facilities is they'll swab it once a week, and they'll send the sample out to a lab that comes back 24 hours to 48 hours later. Too long in some cases to catch the outbreak. The Sample 6 scheme, with this engineered phage that had originally gone for other clinical applications, they can do it in 3 to 4 hours. So they can actually do it on site. Right now, they're in multiple industrial partners, including Legal Seafoods, testing out. They did a soft product launch 2 months ago, and they're planning a serious product launch in the beginning of 2014. So we're very excited about how this system that actually got pushed instantly by interest of investors, I think, is in the end addressing a very important societal problem. I want to shift gears slightly. So all that I've been talking about is really on the microbial level. Looking at bacteria, we also do work in yeast, and this is really where most of the action is in synthetic biology. And the reason is these guys grow fast, and they're easy to manipulate genetically. So as an engineer, you want to build something, test it out as fast as you can. These guys are pretty good. But as many of you can appreciate, many of the interesting problems that a bioengineer or a pre-med student here at Holy Cross would be interested in deal with humans. So the question is, can you go from these lower organisms, can you move up the food chain to higher organisms? Could we reprogram my organic chemistry teacher, Frank Velaccio? Not that we'd want to reprogram Frank, but could you re-engineer mammalian cells, let alone human cells? Yeah. Science, informed consent first, Frank. So briefly what we did is we first wanted to build a mammalian toggle switch. 2 interesting points to that. One is everything that those molecular biologists told us that we'd have problems with did not pan out in the bacterial system, but panned out in spades in the mammalian system. We burned through 2 postdocs. They could not get this to work. And then Tara Deans came into our program. She had done a 3-year 3 years of a PhD program in cell biology at Harvard, decided she wanted to be an engineer, joined BU, which has this transformative program that allows you to make the transition. She came into my lab and says, Jim, I want to try this. And she dug in and just briefly said, this toggle's not going to work. We can't get tight enough repression. That is, it goes back to what the biology says. You can't shut that gene off tight enough. So just briefly, she says, I want to make a really tight switch just as a half of a toggle to shut stuff off. I won't go through this complicated system, but she basically can use a protein, and she can also use an RNA element in case any protein gets, or mRNA gets made, she can knock that down. And briefly, she got this to work beautifully. I'll just, for interest of time, just go that compared to control, she got greater than 99.9% knockdown with her switch. Importantly, you don't only want to shut something off, you want to also turn something on. She could turn this on at a high level, and she could actually have it as a rheostat. Right? So a rheostat could go from a true off to a very high level. So it's like a dimmer switch in your home or in your dorm room. Right? So this is when you're preparing for exams. This is when Valentine's Day comes around. Right? So this is romantic. She can be both very low and very high. And like on many things I shared, the beauty of synthetic biology is you can be highly modular. She can pop in any gene or any on switch of interest. And what she did just briefly is a great kind of demo of machismo for, again, the strength of her switches. She used it to control diphtheria toxin. This is a protein that, when expressed in a single molecule, which will kill your cell. She kept it in the off state, grew these cells up 6-7 weeks, and then could selectively kill off the cells of interest. And this switch is now being used in over 1,000 labs around the world, and people are using this for 2 applications, this switch part in particular, both as a mammalian safety switch to kill off stuff you don't want, but also to create interesting animal models. You can now envision having this under a certain cell type or tissue type, grow your animal up. So if we're going to study aging to make Frank younger, can you model age-related decline by killing off certain aspects of the mouse and then study the effects? So really very interesting. Well, I know there's a lot of interest in stem cells on this campus. We had interesting discussions over dinner. I just briefly want to touch upon 2 applications that we got engaged with. So I know there was a town hall meeting, I think within the last year, discussing stem cells and the ethical quandary that the field was in going back about a decade ago, where then we were largely limited to embryonic stem cells. And the ethical quandary was around how do you get those, and then what should you be allowed to use them for, and there were significant federal regulations. Well, those pressures eased about 7 or 8 years ago when Yamanaka and others discovered that you could take adult cells out of a person, hit them with 4 different proteins, and reprogram those cells from whatever mature state they become back to what's called the induced pluripotent stem cell state. And the idea now, the hope is, this is more, you could take different cells out of somebody's body, make them into what you need to, say, replace different things that Frank is feeling he's lacking, and put it back into Frank or somebody, and have them rejuvenated or address disease. We're very far away from the actual applications, though you'll hear crazy things in largely outside the United States. But the biology is beautiful. One of the challenges is this methodology of taking the cells out is very slow and inefficient, typically involving viral vectors. So you'll typically use a virus to get the genes into the cells to turn on the proteins you want. But this can actually cause problems. You have now stuff being inserted into the genome, which in some cases can lead to cancer. What we did to address this was work with George Daly and Derek Rossi. We took the virus out of the equation, where now you could deliver precursors directly of the protein, mRNA, into the cells and have them become reprogrammed. Much more efficiently, 100 times more efficient, and in just 2 weeks. And we could then also redifferentiate those into cell types that we want. Further, with George's group, much of the work up till then involved fibroblasts. You have to basically take a sample, kind of go into a muscle or skin, to take that out of person to make it into stem cells. With George's group, we used synthetic biology and systems biology to show you could take or create stem cells out of peripheral blood. So just as, say, now diabetic would take some blood to make a measurement, you could now, in principle, take blood and create readily stem cells. Well, again, from an ethical standpoint of you're not really sure what you're doing in terms of what its impact. Around synthetic biology, some of you may be aware there's a DIY biology movement, do-it-yourself biology, in this country and now really broadly around the world, where people are buying cheap equipment to put in their garage, to put in their kitchen, or they're building cheap equipment to put in their garage, to put in their kitchen, to do molecular biology experiments. Interesting, I think, as a hobby. It's being pitched as cutting-edge science. I don't think that's cutting-edge science, but one of the proponents of that, Rob Carlson, wrote a piece in the New York Times highlighting our 2 studies, putting them together and say, as a result of the work from George Daly and Jim Collins, now it's feasible for somebody to order the needed mRNA from a company. You can order this, they'll send it to you. take their own blood and make their own stem cells for research purposes, personalized medicine, or own medicinal purposes. And I was like, oh, that is not what we intended this for. And it actually caused some interesting problems for us with some funders. We can come back. Well, last set of examples that I want to talk about is an area that for those young people here who are thinking about exciting areas to get into, I really encourage you to look at the human microbiome and broadly microbiomes. So the human microbiome is this notion that we are not alone in our bodies. We have 10 to 100 times more bacterial cells inside our bodies than we do mammalian cells. Right? So the phrase, that guy bugs me, has now an additional meaning. It's only about 3 to 4% of our mass, but we're beginning to appreciate more and more the big role that that microbiome plays in disease and in health, whether it's weight, whether it's allergies, bowel, et cetera. We got excited about the idea that you could use synthetic biology to re-engineer the microbiome to address a range of issues. So with support from the Bill and Melinda Gates Foundation, we're developing engineered probiotics to detect and treat cholera. It's a very nasty intestinal pathogen. And what we're doing is taking lactobacillus. So for those of you who eat probiotics in your yogurt, this is the probiotic typically in your yogurt. We are endowing those bugs with decision-based circuits that are going to respond to 2 signaling molecules given off by cholera. If they sense it, it's an and logic gate I referred to earlier. If they see both, they flip on and they produce an antimicrobial peptide specific to cholera. The advantage for this is you can now spike it into yogurt or into a pill in the face of a cholera outbreak, say, for example, in Haiti on the heels of the earthquake. And why that's important is these drugs are very expensive. This would not be. And 2, they can be very toxic. And this, in principle, if we get it to be tight enough, would only express the potentially toxic peptide in the individual who is infected. It's working very nice in a dish. We're moving into mice in the coming months. And we've actually now got strong support to extend this to other intestinal pathogens and interest from Lifebuoy, which is the brand in Unilever that develops hygiene products for Asia, Africa, and Latin America, where they want us to explore how you could introduce this in the skin microbiome. Our hands, every part of the skin is covered with different bugs, some of which can cause real problems. The second and last is that we're also interested in directly re-engineering the bugs in your gut. So for those of you who take probiotics, I hate to break it to you, but for the most part, you're wasting your money if you're actually buying it for the probiotics. They really do very little. They maybe change the expression inside your gut, a little bit of some of the genes. 3 things will basically alter your microbiome: disease, travel to an exotic place, and fiber 1 bars. This is a study out of MIT. So if you can have one of those as a precursor, then maybe you can have an impact. But for the most part, the probiotics will not alter the makeup of your gut. And even with the synthetic one I showed you, there is the possibility they're not going to actually stably integrate into the microbiome. And that's fine. Just keep taking the yogurt or the pill. Okay? And provided they're there to do their thing, can help. Again, most are taking probiotics that think you're changing, you're eating the good bacteria. It's incredibly stable. We're recognizing that. So we are now launching, and have launched, is a product to do cell or gene therapy for the bugs in your gut. We want to deliver circuitry to the bugs in your gut to address things like lactose intolerance, to address things in the lung for asthma, to address irritable bowel syndrome. And we're using bacteriophage. So this is the virus for the bug, whereas you'd use a human virus to get for gene therapy for human cell. We're using the bacterial virus, delivering cargo, and we have shown already that that delivered cargo works as well as if you transfected it with a plasmid, kind of a ring of DNA, which is the typical way. And we now have just launched animal studies on mice to see if we can get it in our, we're rapidly moving this and actually early in discussions with a venture firm to explore this as a possible startup. With that, I just want to show I've had a chance to really work with an amazing, interesting set of networks. This is the one I'm proudest of, a network of outstanding young people that are in my lab. I don't have up there, but I have Jonathan Yao, who is a recent grad of Holy Cross, just joined my lab as a research scientist, absolutely first rate, not unexpected. He was incredibly well trained here at Holy Cross, very broad-minded guy who's already impacting our antibiotic studies. So with that, why don't I end and we can open up for questions and discussion. Don't be shy. Yeah.
How do you deal with mutation, naturally occurring mutation in the things that you've already engineered?
Yeah, it's a great question. It's a big problem. The bugs generally don't like what you put in, so you're going to generate stress responses that are themselves mutagenic. They're going to alter what you put in. we're dealing with it in a few things. And I'll make then also an anecdote about the President Obama group, the Bioethics Commission. We're trying to engineer non-mutator strains, so to reduce mutation rate. And we're exploring how you can have robust circuit designs so that you'd minimize the impact of mutations. Third is we're trying to actually look to see small short cycles of use. So wherein you wouldn't actually have full integration for too long, you'd have to replenish. What was interesting on the Obama Bioethics Commission, they kind of pressed us on the same point. And you had two schools of thought, the kind of the uninformed school, which was the concern that something is going to mutate and become something worse than you thought. And that has never happened in our lab, and I'm not aware of groups. That it's usually loss of function. And interestingly, so the informed groups of members of the commission were nonetheless very concerned. They said, okay, if you're going to do this and it's going to have a practical impact, how are you going to ensure that what you put out there is going to be able to still do what you want? So we're trying to do it from an engineering standpoint. Yes?
Have you explored any epigenetic switches, like protein fold or DNA methylation or anything like that?
Yeah, so that's a great question. The field has not published any work on either of the schemes you have. We have now a methylation-based switch. So we're doing chromatin modification and laying down methylation patterns as a means to get higher levels of control, particularly in eukaryotes. And so right now we're doing it in yeast, but we're hoping to move in mammalian cells. So actually, you were the protein guy, right? So where the field actually has a huge open gap right now is people are not developing protein switches. Right? So the switch I showed you to start off was a transcriptional switch. It's about 20 minutes to get machine or transcription translation. Our RNA switches can get you down to 5 minutes. But you can envision with protein folding or protein phosphorylation switches that you can be in that tiny fractions of seconds. And for whatever reason, the field has not moved in. I think it's because we don't have enough chemists and in the field. It's largely was the physicists, the engineers, computer science came in, biologists came in now, and the chemists are just coming in. And I'm hopeful that we'll see some new developments on that front. Yeah.
When you started being talking about the flip-flop
Yeah.
model, and have you seen in either the connections you mapped out that are naturally present or maybe indirectly any examples of that kind of switch that's naturally occurring where one gene is either expressed or not, and there's some benefit to that?
Yeah, it's another really interesting question. Yes. So in fact, the lytic lysogenic trade-off or switch that I talked about for the bacteriophage, in fact, is in essence a toggle switch. And it's one that evolved a billion years of sorts. Other labs around the country have actually identified multiple toggle switches. Interesting, not so much in bacteria, but in higher organisms. So it's actually quite common in mammalian systems to see now toggle switch controls. And in some levels, it's actually even an abstracted switch, and that is they can reduce it down to a toggle dynamic, and yet it might involve 5 different genes interacting in multiple positive and negative feedback loops. Yeah.
What do you consider to be the biggest roadblocks of progress? Would it be wet lab technology or computational tools?
Excellent question. So I'll answer it with, that's not a direct answer to your or question. I think our biggest roadblock is lack of biological knowledge and mechanistic insight into how these things, into how the different components function together. So what I didn't share, I gave a little bit, is it is incredibly hard to get any of this stuff to work. So it's relatively straightforward for us to map out our schematic. We've gotten pretty good as a field to do the computation. Generally, qualitative modeling is first rate right now. Quantitative modeling has been slow. We don't have enough characterized parts, well-characterized parts to pop in. If you're an electrical engineer, gadgetry guy, there's literally billions you could buy. But where it breaks down is when we start putting this stuff together, all bets are off on properly modeling the behavior of that to quantitatively predict how it would function. Yeah. It's part specific. So for example, on a promoter, you would, for example, look at its inducible range. You might analyze its expression dynamics. There's a lot of talk around this in the field on so-called standardization needs. And say, well, once we get standardized, and I actually think it's a false notion on 2 counts. One is standardization is really not a central engineering element. We don't teach it. It is an essential industrial element, right? You want, if you buy an 8-inch screw from China, you want to do the same one that you'd expect from Italy. And the second is I think standardization goes out the window in biology in the sense that For those who've been in the wet lab, your media may be different from my media. Your strain may be slightly different. The temperature, my bugs would love this room right now, but my lab's not this temperature. My lab's 30 degrees cooler than this room, right? But you've got even slight temperature changes that can impact. And so you have all of that in that right now we just don't know enough biology to properly engineer biology in the way we'd like. Yeah.
As a biologist and not an engineer, do you think part of the problem of trying to characterize parts is that the parts are too multifunctional? So if you look at particular proteins, that those proteins have too many potential applications for you to be able to characterize them in isolation, or even in simple contexts. Do you know what I mean? That,
yeah.
It totally depends what's around them. It depends on their environment. It depends who they're interacting with. So therefore, a simple characterization that you could plug in as a part becomes much more difficult.
Yeah, I think that's fair. And I think it's an overlooked point by many engineers. And a few points to that. One is I think we're constraining ourselves of thinking too much as engineers and bringing engineer analogies and not appreciating all the odd things that can happen with biological systems. And in particular, it may function well in vitro, so even outside the bug. You put in the bug, well, you're not going to get rid of the host machinery. It interacts in ways you just can't predict. But even on that, a point to share is engineering is hard. So to build the circuits you'd get from your little kid at the Museum of Science in Boston, fine. Yes, we can model that really well, little clip. For analog devices, the circuits you put in, they cannot model those. So once you get to a large number, it's all bets are off. We actually had support from analog devices about 13 years ago before I moved to molecular biology, where they challenged us, says, Jim, we're making these large circuits. We have no idea how they're going to function. We're trying to find notch filters for A to D converters, right? So where you're going to take the Indigo Girl song, put it on a CD, all of which are probably dating me in an incredible way in this room. You want to kind of chop out the hiss on that conversion. We want to do it really effectively. We don't know how to do it. And they actually turned to us to see, could you model it on the basis of the brain? And we had an incredibly talented guy from Ray Stata's group come and work with us. But it was interesting for us where they says, what we do? We make 1,000 of these with variants, and we try to find the one that works the best. So it's as if they're doing, it's not directed evolution, but generate diversity and selection for the one, even in one of the better engineering firms in this country. Sorry, Dr. Lindbergh.
Yes, a fairly silly question, perhaps. Even back in the day, we were worried about bioterrorism, and it seems like with these kinds of parts off the shelf, it might be even easier to engineer for malign purposes. Are there any regulations associated with acquisition of the parts?
Yes. So this is a big concern. And the DNA synthesis companies have gotten together where they screen the requests, and they will share what the requests are coming in. So you can imagine a clever person chopping up a polio virus or SARS virus into 12 different schemes, send it to 12 different ones, and then put it together. And so there's a focus on that. I worry less about that. So I've been at many FBI meetings in synthetic biology, and they've been in my office more times than I'd care for on this topic, to just about terrorism. And I keep telling them that I'm not worried about the engineered strain. I'm worried about the natural strains, that nature's going to get us, and we're not ready. And I was saying at dinner, we're potentially moving toward a post-antibiotic era. Forget about the terrorists. The bugs are going to get us, and we are not in a strong position to respond. And they nod their head yes, but they don't handle that aspect. They go after the bad guys. But I'd say there are other bad guys that we really should focus on, because it's incredibly hard to work on these. The idea that you've got an 18-wheeler truck bed that's got a terrorism lab that's driving around the desert is completely ludicrous. For anybody who's worked, right, the slight contamination will mess up your lab. So you run around the desert in a truck bed. My goodness, I've shut down my labs for 3 months at a time when a kid brings in something he shouldn't. So there are people thinking pretty nasty things about this, but I'm much more worried about our organic enemies out there. More questions? Yeah.
You're talking about the biofilm busters, and
Yeah.
One question I have is, like, once they're inserted in the organism, do they always remain there? And if so, do the sensors kind of get disrupted over time, or is it just like a constant process that goes on?
Yeah, that's a really interesting question. So the lysogenic ones that would go in, not for biofilm busting, for antibiotic boosting, will tend to just stay there. And they'll put very low evolutionary pressure on the bug. The biofilm busters will not. If they do their lysis, they're getting out. And now what will happen is they need another bug to propagate. And interestingly, I think phage are actually the most abundant microbiota on this planet. So if you look, they constitute the most DNA on the planet of the phage. So the nice thing on that is that once they run out of bug, they're done. The problem with some of the therapies that we think about, say in the gut, and that's why we're focusing on the surface, is worried about immune response. And the fact that phage will also trigger immune responses, so to speak, in the bug. So the bacteria, when they see a phage infection, will start spitting stuff out. They'll drop their plasmids, they'll drop antibiotic resistance genes. So the concern is that, as often happens, we may be making the matters worse. And it's something we need to analyze, and regulatory groups need to look at more carefully. But it's a very interesting point. More questions from students. You look at your student? We're really young faculty here, student. More student questions. Come on, I'll call on somebody. I'm a regular professor, and I tell my class, do not look away. If you look away, I will call on you. You're a holy cross. Everybody has something interesting to say. You wrote all interesting essays to get into school. Somebody has to have a question. Yeah, thank you. Yeah, so in fact, that's specifically, those two areas are lab focus areas for some of the labs that are using this switch. So the idea is that they're actually looking to kill off certain cell types, in the case you bring up, certain neurons, to now study what happens with neural degeneration. We're involved with another lab that's looking to kill off certain peripheral neurons, to go back to the example that I started with on the balance, wherein you actually have significant loss of sensation, both from a threshold standpoint, but also you start losing neurons when you're older. And that actually underlies balance difficulty. So there's a lab that's focusing on that, where they can use this in a targeted fashion to knock out certain neurons at a certain development stage of the mouse. Any more questions? Yes?
Just like I heard you mentioned earlier about the DIY bio.
Yeah.
Things with biohacker spaces. I've been kind of following up on that stuff lately, and I just want to know, like, what do you think about it? Do you think it's going to cause problems for like people if they manipulate certain things? I know you said earlier you're not worried about bioterrorism, or do you think this would push something towards like educating the public or going to the cutting edge of science?
Yeah, it's interesting. So I'm maybe the only public critic of DIY biology. So I have spoken a fair amount about this because there are many public proponents of it. And my criticisms are multifold. So I'm not concerned that there's going to be cutting-edge science. And there's many reasons for that. It's not that because they're amateurs. So synthetic biology was launched by amateurs, including myself in molecular biology. It's that they're under-resourced, both in terms of proper equipment and time. So to make significant advances as a hobby on a Friday night, you're going to play with it, I think is a pipe dream. That to actually make an advance in science, you've got to show up, and you've got to be in there working very long hours, particularly on the engineering side, because it takes so long to get there. And as a result, I basically said that at best, many of these groups are going to make a mess in their garage or kitchen, or get themselves or their neighbors sick, because they're ordering bugs that can make you sick and then not proper use. What I do like about the DIY biology movement is two things. One is I do think it's useful as a hobby for somebody to learn about molecular biology. So my daughter's 14 years old. She's very into science right now. She got a chemistry set for Christmas, and we do experiments, both that are in the kit and that we make them up. And it's a lot of fun. We have it in our dining room. I don't think I'm advancing chemistry. I was taught by Professor Vlacho. He showed me what real chemistry was. But we're having fun. And from that in DIY biology, I can see it. The other, which is very positive, is there are now very well-resourced labs that are popping up. Brooklyn has one. I think San Francisco has one. Boston's putting in one. So now if you have somebody they can put in the time commitment, now it's possible you could see some interesting developments that could come through that. But I bet you dollars to donuts, there's not a one that's putting in the time commitment you'd expect. My students, postdocs, have put in 40, 80 hours a week, and it's taken them 2 to 3 years to get something to work. And so it gives you a sense of the amount of time you have to commit to see the finding. So, and yet the media, you has just latched onto this, and there's story after story. This is, the democratization of science. We've got cutting-edge work, and I keep pressing them. Let's wait till we see a discovery come out of one of these before you play them up so much, versus just an interesting hobby like me with my daughter and my chemistry kid. Yeah.
Do you think it's plausible to develop a system that reestablishes properly functioning genetic line, like say in cancer cells or something, you can develop a way to reestablish apoptosis and lines?
So there are groups trying to do this, and I think they've done a decent job in a dish with cell lines. And then the challenge becomes, how do you actually translate that into an in vivo model, so you can start an animal, let alone into a patient. And this is where we run up against the challenge of the complexity of the system. That, I think to challenge you all, you take these beautiful courses here at the Cross, these large textbooks, and you have a sense where there's just so much out there. I maintain we know very little, and which is both a challenge and a great opportunity for you and still for me to try to figure out how all these things function and work together. And it's why I think most stuff fails. As the professor said earlier, these things have these multiple functions, and how do you capture them all, and how do you predict what's going to come up? And this is just on one protein. And now you think of the thousands inside, let alone the many, many different cell types. And it's why so many of these cool ideas that I challenge you all to think about and come up with and pursue really seem to, unfortunately, break down when you get to translation. And I'm hopeful that as more engineers, more physicists, more liberal arts trained scientists come in that can see the big picture, we'll begin to kind of embrace the complexity, not view it as a bug, view it as a feature, and see if you can harness a little bit of it to try to make better inroads. But your idea is a very good one. And yeah, there have been in vitro demos of success, but not yet in vivo. Well, recognizing the heat stroke threshold of even healthy 20-year-olds and respecting my older colleagues who are here, why don't we end there? And I want to thank you all for coming out on this Wednesday night.